Video we'll see the definition and some examples of homopolysaccharides. Now coming to the definition, homopolysaccharides. Homo means same, poly means many, and saccharides means sugar units. So these homopolysaccharides are made of many sugar units which are of same kind. Homopolysaccharides are made of only one type of many monosaccharide units. All the sugar units that are present in homopolysaccharides are of same type. Some examples of homopolysaccharides are starch, glycogen, cellulose, dextrin, inulin, and chitin. First we'll see about starch. Starch is the storage form of glucose in plants. For example, in potato, in grains, in seeds, etc. So this starch is mainly composed of two constituents, that is amylose, which makes of 20% of starch, and then amylopectin, which makes 80% of starch molecule. This amylose is a linear polymer of D-glucose units. That is, the structure of amylose is it has glucose units linked linearly, and the bonds which link these glucose units are called as alpha-1,4 glycosidic linkages. And another molecule or constituent of starch is amylopectin. Now this amylopectin is a branch polymer of glucose units. So in addition to linear linkages of glucose units, this also has branches. Now this peculiarity here is the branch bond. The bond that is present near the branch is alpha-1,6. So in the straight chain, the linkages that are present between glucose units are alpha-1,4, whereas the bonds that are present at branching point are alpha-1,6 glycosidic linkages. Glycogen is the major storage form of carbohydrate in animals. So it is often called as animal starch because similar to starch, which is the storage form of carbohydrate in plants, glycogen is the storage form of carbohydrate in animals. So glycogen is found mostly in liver and also in muscle. The structure of glycogen is similar to that of amylopectin, that is the branched molecule of starch, except that the difference between starch molecule, that is the amylopectin of starch, and glycogen is that glycogen is highly branched. So this glycogen has a branch point after every 6 to 7 residues meaning glucose units, and each chain has about 13 to 14 glucose units. So there is a branch point after every 6 to 7 glucose units, and each chain will last for about 13 to 14 glucose units. As discussed, glycogen is present in muscle as well as in liver. So muscle glycogen function is to act as a readily available source of glucose for muscle, whereas liver glycogen is mainly concerned with storage and maintenance of the blood glucose levels. So whenever muscle needs more of glucose, muscle glycogen acts as a source of supplier of glucose, whereas liver glycogen is mainly concerned with the maintenance of blood glucose levels. Next important homopolysaccharide is cellulose. Cellulose is the most abundant carbohydrate that is found in plants. Now, unlike starch and glycogen, this cellulose is an unbranched polymer. That is, it has long straight chains, and now these straight chains are joined together by beta-1,4 glycosidic linkages. If we see in starch and glycogen, the straight chains were linked by alpha-1,4 linkages, but in cellulose the straight chains are linked by beta-1,4 glycosidic linkages. Now cellulose does not have any calorific value in humans as it is not digested because of the absence of the enzyme cellulase. But ruminant animals have bacteria which produce cellulase that digests the cellulose. So in ruminant animals, cellulase is present. So they can digest cellulose. Nutritional importance of cellulose in human beings: as such, for humans, cellulose has no nutritional significance, but cellulose is a component of fiber in the diet. So, even though there is no metabolic requirement for fiber, high fiber diet is associated to reduce a number of diseases like cardiovascular disease, colon cancer, diabetes, and diverticulosis. So, this cellulose is present in unrefined cereals and it increases the bulk of stool, helps in intestinal mobility, and also acts as a stool softener, preventing constipation. So, even though cellulose does not have any nutritional significance, presence of cellulose helps in diet, helps in reducing a number of diseases like cardiovascular disease, colon cancer, diabetes, and also acts as a stool softener, preventing constipation. Next important homopolysaccharide is inulin. Now inulin, it is a polymer of fructose. Till now we have seen that starch, glycogen, and cellulose, all of these were units made of glucose, whereas inulin is made of fructose, and that too it is linked by beta 1 2 glycosidic linkages. It is present in tubers of some plants like chicory, bulb of onion and garlic. Inulin is hydrolyzed by this enzyme called as inulinase, which is not present in humans, and so it is not utilized as food. But there is some importance of inulin. Inulin has clinical importance because it is used in the studies of glomerular function rate or in the kidney function tests. Now, why inulin is used as a marker for GFR, that is glomerular filtration rate, is inulin is filtered but not reabsorbed and not secreted. So, whatever inulin that is given to the patient, it has to be filtered by the glomerulus and it has to be excreted outside, and it is not reabsorbed. So the amount, if we are giving for example 50 milligrams of inulin, entire 50 milligrams of inulin should be excreted outside, and this helps in telling about the filtration capacity of the glomerulus. So that is why inulin is a classical marker for glomerular filtration rate, and it is used in kidney functioning tests. Next homopolysaccharide is chitin. It is present in the outer skin or the exoskeleton of insects. Here in the exoskeleton of insects and in crustaceans, it is the second most abundant polysaccharide in nature after cellulose. Now it is made of N-acetyl glucosamine monomers. Till now we have seen glucose and fructose. Now chitin is made of N-acetyl glucosamine, which are joined by beta 1,4 linkages. So firstly we have seen starch in the homopolysaccharide. Now starch has amylose and amylopectin. Amylose has alpha 1,4 linkages in its straight chain, whereas amylopectin has alpha 1,6 linkages in its branched chain. Similar to amylopectin, there is glycogen, which is a highly branched structure. And then we have cellulose. Cellulose is not branched, it is a straight chain structure which has glucose units, but these glucose units are joined together by beta 1,4 linkages instead of alpha 1,4. And then we have inulin is another homopolysaccharide which is made by fructose units. Now these fructose units are joined together by beta 1,2 glucose units. And finally we have chitin as another homopolysaccharide which has N-acetyl glucosamine joined together by beta 1,4 linkages. Thank you.